Hi, it's Miss Vitale. This podcast is the first of two on microbes, and this one focuses on viruses, which corresponds with chapter 19 in the textbook. In 1883, Adolf Mayer, who was a German scientist, discovered that a tobacco plant disease could be contagious when he sprayed the sap of an infected plant on a healthy plant. But he couldn't find a microbe in the sap, so he thought the disease was caused by a very small bacteria. 10 years later, Demitri Ivanowsky, who was a Russian, filtered the sap through a filter that removed bacteria, but the sap still caused tobacco mosaic disease, so he thought maybe the bacteria produced toxins that were able to pass through the filter. In 1897, Martinus Beijerinck, who was a Dutch botanist, discovered the stuff in the filtered sap could reproduce, eliminating the theory of a toxin, but the infectious agent could only reproduce in the plant, not in petri dishes like bacteria. In 1935, Wendell Stanley, an American, crystallized the infectious particle now known as TMV, tobacco mosaic virus. The term virus is Latin for slimy liquid or poison. Stanley's discovery seemed to imply that the virus was just a large molecule, but it was clearly replicating itself. A virus is a nucleic acid core surrounded by a protein coat. Most viruses don't cause disease. They are relatively harmless, and some introduce useful genes. In the 1940s, scientists could see viruses using electron microscopes. The biggest viruses are the pox viruses. They're about 250 to 400 nanometers in diameter. A nanometer is 1/billionth of a meter. Polio is one of the smallest, about 20 to 30 nanometers in diameter. Remember that viruses are extremely small, much smaller than bacteria. The nucleic acids of viruses can be double-stranded or normal DNA, single-stranded DNA, and even double or single-stranded RNA. The capsid is the protein coat of the virus. It comes in all different shapes. Many viruses that infect animals, like influenza and AIDS, direct the host cell to make a viral membrane from the host cell membrane, together with their proteins that are specified by the virus's genes. Some viruses have extra structures that help them infect the host. Viral envelopes cover the capsid. They are structures to help the virus stick to the host. Viruses have many of the properties of life. They are highly organized, they reproduce, they respond to their environment, and they evolve. But they cannot obtain energy from their surroundings, they can't perform metabolic reactions, and they can only reproduce by using the biochemical machinery of a host cell. Viral reproduction. Most viruses reproduce using the following stages. The virus attaches to the host cell, and then it injects its nucleic acid into the cell. The cell makes proteins specified by the virus's genes. Then the cell replicates the virus's DNA or RNA. The new proteins and nucleic acids made by the host cells assemble into new viruses. The new viruses are released from the host cell. A given kind of virus can only infect certain kinds of cells. Viruses are usually adapted to very specific hosts. For example, cold viruses and HIV are specific to humans. This is called species-specific. So how does a virus attach to a host? Specific proteins on the surface of the virus bind to the receptor molecules on the surface of the host cell. The attachment site may be on all of the host cells or only on a special set of cells. For example, cold and flu viruses attack respiratory cells. Pox viruses attack skin and nerve cells. Polio viruses first enter the digestive tract, then they move to the blood, and they gain access to the cells of the body. About 1% of the cases of people infected with polio have their nerve cells infected, which then causes paralysis. Once a virus is attached, its nucleic acid enters the cell in many ways. Enzymes of the virus digest the cell wall. Endocytosis, where the cell membrane enfolds the particles, is one way, and the flu viruses enter cells that way. Viruses are oftentimes enclosed in bits of the cell's own membrane, which they capture when they leave the infected cell. HIV does this. This stolen membrane then fuses with the membrane of the next host cell. Once inside the cell, the virus genes direct the synthesis of new viruses. There are two different methods, the lytic cycle and the lysogenic cycle. In the lytic cycle, the cell lyses, which means it breaks open. Bacteriophage's whole cycle during the lytic cycle is only about 20 minutes, and this produces over 100 new phages. The first thing that happens in the lytic cycle is the virus attaches to the host. Then the virus DNA is injected into the host. The host's DNA is hydrolyzed or broken up by the viral DNA. The virus DNA directs the host to make new cell viruses, new virus parts. Then the virus's direct production of lysozyme, which is an enzyme that digests the cell wall, causing the cell to break open, and the new viruses are released. Each one of them then go on to infect another host, and the cycle continues. Viruses that use the lytic cycle are called virulent. Viruses in the cells they infect are constantly mutating. As the cells evolve to resist the viruses evolve to infect the new types of cells. For example, every year there are new different strains of flu, therefore new different vaccines have to be produced each year. The lysogenic cycle does not destroy the host cell immediately. First, the virus attaches to the host and injects its DNA. Then the DNA becomes part of the host. The cell reproduces with the prophage. The prophage is the DNA with the viral DNA in it, and it passes the prophage on to the daughter cells. All of the new cells are infected with the prophage, meaning they have a piece of the virus. The viral DNA may leave the DNA of the host and enter a lytic cycle. At that point, the lytic cycle begins, and all of these new cells that were created with the prophage in it now enter the lytic cycle. New parts of the viruses are produced, and the viruses burst out of the host cell. A virus is the most elaborate of the mobile genes, but there are other types. Most lack an exterior coat or membrane, but like viruses, they have DNA or RNA that induce the host cell to make copies of their genes. A plasmid is a small piece of circular DNA or RNA in a bacteria or yeast cell. They include anywhere from 3 to 300 genes. They replicate autonomously, meaning that the genes are separate from the DNA of the host cell, and they can be replicated even when the host DNA is not replicated. A cell can contain as many as 1,000 copies of a single plasmid. Sometimes bacteria reproduce using conjugation. Two bacteria cells temporarily attach to each other and exchange genetic material. Conjugation is how plasmids get around. Plasmids may contain genes that benefit the host. For example, R factors are resistant factors. That means the plasmid contains genes for enzymes that make them resistant to things like antibiotics or heavy metal poisoning, or enzymes that metabolize components in oil spills. Molecular biologists have synthesized recombinant plasmids that are not found in nature. Transposons are mobile DNA sequences that can replicate only when it is part of the host cell's DNA. Jumping genes that move themselves to new locations or duplicate for repeated insertions elsewhere are what transposons really are. Viroids are small circles of RNA that infect plants. They interfere with the plant's metabolism and stunt the plant's growth. They can kill plants by interfering with their growth and development. Prions are infectious proteins. They may be an improperly folded protein that converts normal proteins to prions. Stanley Prusner was an American scientist who won the Nobel Prize in 1997 for his hypothesis of prions. Mad cow disease is caused by a prion. Even though viruses and mobile genes are not alive, they are still independent entities whose replication is subject to natural selection. A virus uses energy from its host and impairs the cell's ability to grow and reproduce. So the host evolves ways to resist viral infections. For example, most E. coli bacteria produce restriction enzymes that cut up foreign DNA. Those restriction enzymes are the same ones that are used in genetic engineering. As hosts develop resistance, the viruses evolve to get past the cell's defense. Viruses that infect E. coli have DNA missing that is targeted by the restriction enzymes. Viral multiplication cycles have evolved to become very efficient. Cycles are rapid and produce large numbers of viruses. There is no junk DNA in viruses, and the number of genes is minimal to make replication more efficient. Viruses probably evolved from their host cells. The nucleic acids of viruses have a lot of genes in common with their hosts. The original source of a virus's genome may have been plasmids or transposons that left the host cell. Many different types of viruses have evolved. Viruses have different types of RNA and DNA, proteins, and may or may not have a viral envelope. As I said before, the viral envelopes are outer membranes made of the liquid bilayer, just like the cell membrane. They contain glycoproteins that bind to receptor proteins on the host cell. The viral envelope fuses with the host membrane and the viral genome enters the cell. Once the host cell makes new virus parts, the virus exits the cell by exocytosis, getting a new envelope from the cell's membrane. Some viruses, like herpes, get their envelopes from the nuclear membrane. The DNA of the virus reproduces inside the host's nucleus. Many viruses contain the nucleic acid RNA. There are three basic types of RNA in viruses. The first type is when RNA is a messenger RNA and it is translated into viral proteins right after infection. The second type is when RNA is a template for messenger RNA. Messenger RNA is first made by the host cell and it is used to make proteins and more viral RNA. And third, there are retroviruses. They contain reverse transcriptase. That's an enzyme that transcribes DNA from RNA, and the new DNA becomes a provirus, which is part of the chromosome of the cell. HIV is a retrovirus. Vaccines are modified or killed forms of a virus that are used to stimulate an immune response. An English doctor, Edward Jenner, in the late 1700s learned that people who caught cowpox from milking cows were immune to smallpox. In 1796, Jenner scratched a boy with a needle that had fluid on it from a cowpox sore. When he was exposed to smallpox, he didn't get it. This was the first vaccine. Other vaccines like polio, rubella, measles, mumps, pertussis, hepatitis B exist, and you have been vaccinated for most of these. Vaccines are our best defense against viruses. We don't have drugs to fight them, like antibiotics. Some viruses can cause cancer. Viruses that cause tumors only multiply when the eukaryotic cell multiplies. Tumor viruses exist independently only to find a cell and infect it. After infection, the genes of the virus become integrated into the host's DNA and stay there. The viral proteins then produced by the cell ruin the cell's ability to control cell division, and the cell divides rapidly, creating a tumor. Oncogenes are viral genes whose expressed proteins interfere with the host cell's ability to control cell division. Most oncogenes are our own genes that have been damaged. Proto-oncogenes code for proteins that affect the cell cycle. Viral oncogenes probably evolved from the cell's proto-oncogenes.